My project was the effect pH levels have on raw meat, and specifically how do liquids with different pH levels affect the bacteria growth on different types of raw meat. Based on my research of buffering capacity, which is basically when cells are able to regulate their pH levels into whatever level is optimum, similar to guardrails on a road, and confounding variables such as polyphenols, I hypothesized that the more acidic liquids will prevent bacterial growth on raw meats because the acidity will deprive the bacteria of nutrients it needs to grow. I reached this hypothesis because I applied the class lesson about how enzymes can be negatively altered by extreme pHs. In my experiment, I had 3 different types of meat and 3 different types of liquid with varying pHs. I divided the petri dishes so that there was 9 in total, each liquid falling into 3 different petri dishes and each meat into each type of liquid. I placed them in a room temperature environment of about 70 degrees Fahrenheit and observed every 2 days. My independent variable was the pH levels, with vinegar at 3, water at 7, and kale juice at 12. My dependent was the amount of bacteria growth, and some controls were the amounts of liquid and meat, temperature stored, a 2-week trial, and how old the meat was when the trials began. These pictures highlight the bacteria growth from day 1 to day 14 of the first trial for the kale group, with the growth spots highlighted. As you can see, each meat expanded in size and they all smelled pungent and disgusting by the end. These pictures show small but present bacteria growth on meats placed in water. These pictures show no bacterial growth on any of the meats placed in vinegar. The first graph shows there was never any bacterial growth on the vinegar dishes. The second graph, my control group, showed how bacteria was able to grow well on steak but not much on the other 2 meats when placed in water. And the third shows that each meat held substantial bacterial growth when placed in an alkaline liquid of kale juice. As you can see, bacteria growth was obviously present on day 14 of trial 2 in the kale groups as well. The trial 2 water pictures also showed minimal growth on the meats when placed in water. And lastly, the meats placed in vinegar showed no sign of bacteria growth. My data from trial 2 reflects that of trial 1. As you can see, there was no growth with vinegar, little for water, and a substantial amount of kale juice. The averages from both trials stay the same, and it is important to note that when there was bacteria, it produced the highest amount on steak each time. A similar experiment conducted by D. Vlahova-Vangalova and S. Dragovev produced similar results with the most microbial growth occurring with a basic liquid and the least growth with an acidic liquid. One thing I would change about the experiment is the alkaline liquid I chose to use. The kale juice was a very similar color to the bacteria, so it made it much more difficult to measure. Some other disadvantages of this experimental design were that only 3 of the 14 possible pHs were tested, and it was a small sample size. However, this size sample also allowed me to observe minute changes from the day-to-day and to control the experiment better. It provided a solid basis for potential future work. These results can be applied to many areas of study, especially medicine. If we're able to control the environmental pH through medicines, we can individualize treatments for specific disease-causing bacteria. Additionally, we can use it to test when we should change the pH environment of plants to achieve a desired effect that carries all the way throughout the production process, from farm to pantry. The trends of bacteria growth on raw meat with liquids of differing pHs were consistent throughout the trials, ensuring the purpose was achieved and proving the hypothesis correct. In conclusion, the more acidic the liquid base, the less bacteria will be produced because the extreme environment will prevent any bacteria from being able to thrive and sustain a presence.